Hey guys, we're back with another video. Today we're gonna do 9700 variant 12 February March 2021. So yeah, the change from next session is that we will have 1 hour 15 minutes instead of 1 hour. So that's a lot of time, okay? So when you test yourself, you can actually give yourself 1 hour 15 minutes or just 1 hour to test your limits, right? Because these were supposed to be done in 1 hour, so why not? But I have feeling to compensate for the extra 15 minutes, they might give a bit more difficult questions, right? Okay, let's begin. Oh, before that, the threshold, right? I'm gonna attach it in the description below. A lot of you ask me for question papers, right? I always attach it in the description, okay? It's in the description along with the threshold, along with the mark scheme and the examiner's report if that's out, okay? So A was at 26, B was at 20, 23. C was at 20, D was at 17, and E was at 14. Okay, so yeah, A star was probably at around, some of you are saying it's 30 since there's a difference of 4, 3 to 4. Or personally, I think it's gonna be at the midpoint between 40 and 26. So around 33, just to be safe. 33 was A star in this paper, approximately, yeah. Let's begin with question number 1. So we have a photomicrograph which shows onion cells. The actual length of the onion cell labeled is 350. What is the magnification? So I can't really do this. I need a scale or ruler. So what you're gonna do is you're gonna find out the ruler, find out the image length with the ruler. Okay. I'm still gonna tell you what it's gonna be like. Like if you do have the printed copy. Wait a bit. It's gonna be around 14 centimeters. When you use your ruler, it's gonna be around 14 centimeters. Now we know that magnification is equal to image by actual length. So magnification is equal to image length 14 into 10 to the power minus 2 divided by actual length 350 into 10 to the power minus 6. Okay, so 14 into 10 to the power minus 2 divided by 350 into 10 to the power minus 6. That's giving me an answer of x 400 times. Okay, so 1 is D. Moving on. Which row correctly shows the presence of a cell structure in typical plant cells and in typical prokaryotic cells? Remember, plant cells do not have centrioles. Okay, plant cells do not have centrioles. So A is wrong. Plant cells do have 70s ribosomes in their chloroplasts. And typical prokaryotic cells, yeah, they also have it. So 2B is right. Why are 2C and D wrong? Prokaryotes do not have cellular cell walls and they do not have lysosomes, okay? They don't have membranous organelles anyway. So 3. Which letter identifies cell structures where semi-conservative replication of DNA occurs? So basically, um, replication of DNA. It occurs in the nuclei. It also occurs in chloroplasts and mitochondria because, according to the endosymbiont theory, they were bacteria originally and they were derived from bacteria. Now they can make their own DNA, they can replicate their own DNA, make their own proteins, etc. So A is correct. Which parts of a cell contain ribosomes? Chloroplast contains 70s ribosomes, mitochondrion contains 70s ribosomes, cytoplasm contains that, but not Golgi body. So 1, 2, and 4 should be the answer. 4 should be C. Moving on to 5. Which range of diameters is typical for prokaryotic cells? I was talking about this in the October-November variant. In variant 1.3. For prokaryotes and for eukaryotes. For eukaryotes, it's typically around 10 to 100 microns, 10 to 100 micrometers. And what about prokaryotes, right? So it's around cell diameters, right? It's around 1 to 10 or 5 to 10. You could say it's around 1 to 10 micrometers or 5 to 10 micrometers. Anything works. So let's see which one works. 1 nanometer to 500. That doesn't work, right? So 1 nanometer definitely doesn't seem right. So that's wrong. Now look at C. It's 10 to 500 micrometer. That's too big. Now this is 100 to 5000. That's also too big, right? So what about B? It's 1000 nanometers. So listen, 1000 nanometers is actually equal to 1 micron. So this is 1 micrometer to 5 micrometer. That looks good, right? That is within this range as a whole. So yeah, 5 should be B logically. Moving on to 6. Which row shows a combination of molecules that could be found together in a single isolated virus? Okay, listen. A virus contains either DNA or RNA. Okay, not both at the same time, either DNA or RNA. Okay, so A is wrong because it shows that it contains both. Now our possible candidates are B or C. No, 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 not C, because in C we also have both DNA and RNA. This is also wrong. It's either between B and D. Now remember, a virus has, out of the biomolecules, a virus has protein coats, right? So it should be D. This is wrong because it shows that the virus does not contain protein. 7. A student carried out 4 biochemical tests on a sample of food. The emulsion was cloudy, fat was present. So A is wrong. Now, Benedicts, this is a trick question. I saw this in October, November 2021 as well. So follow the trend, okay, to do good in your exams on 9th June, right? You need to make sure that you do 2018 to 2021 and also Feb March 2022 along with the specimen paper 2022. Okay, I'm gonna do the specimen paper as well very soon. Okay, before your exam. I'm not sure about other years, so I also have exams coming up. So I'm gonna be a bit busy starting from tomorrow. I'll still try to do my best, alright? So make sure to support the content. It's gonna help me a lot, right? So hit the subscribe button. So Benedict solution, it shows us the presence of a reducing sugar, but not glucose. How are you sure that it's glucose? Okay, we're not sure. Now Biuret solution turns purple, so protein is present. That's true. This turns yellow. It does not turn blue black, so starch is not present. D. D is wrong. Okay, 8. So C was the answer. 8. Which diagram represents a molecule of alpha glucose? Guys, you need to memorize the structures of alpha and beta glucose. Remember, this is what alpha looks like. So in alpha glucose, the OH for carbon number 1, right, should be directed down. It should not be directed up. Okay, that is why C is not a possible answer. It's between A, B, and D. Now, how are you gonna figure it out? What's the trick actually? So remember, it's down, down, up, down. Okay, that's how I remembered it. Down, down, up, down. Carbon 1, 2, 3, 4. Down, down, up, down, up. So that look, down, down, up, up. That's wrong. Down, up, down. That's wrong. It's down, down, up, down. Okay, it is A. Please memorize the structures of alpha and beta glucose. It's in your course book. It's in your course book, chapter 2, right? I hope everyone follows this. Here you go. Wait, where is it? Here. Down, down, up, down. And for beta, it's up, down, up, down. It just alternates, right? Okay. Moving on to 9. Which statements about glycosydic bonds are correct? Condensation between glucose and fructose uses water and forms. No, no, condensation does not use water. It gives off water. One is wrong. One is wrong. It's between C and D. Glycogen contains 1,4 alpha and 1,6. True. True. The hydrolysis of amylopectin uses water to break. This is what I was talking about. This is true. Hydrolysis requires water. True. 3, 4. Amylose contains beta. No, no, that's for cellulose. It should be 2 and 3 only. C is correct. Okay. 10. A triglyceride has ester bonds formed between fatty acids and glycerol. Okay. So ester and glycerol. 10 should be C. Really important MCQ. 11. The diagram shows a phosphated molecule divided into 3 regions, X, Y, and Z. R in region X represents a range of possible chemical groups. What's the question? Region X, Y, and Z affect the properties of the cell surface membrane in different ways. Which row shows the effect of each region on the properties of a cell surface membrane? increases the permeability of the hydrophobic region, repels polar molecules, attracts water molecules. Okay, so remember, to repel polar molecules, you need to be hydrophobic. Could I clarify? So what's the hydrophobic portion of a phospholipid? Remember, a phospholipid has a phosphate head which is polar, and two hydrophobic tails. So Y and Z are actually hydrophobic, so they actually repel water. Okay, Y and Z are hydrophobic, so they repel water. It's between B and D. Now remember, X, the phosphate head, it's actually polar, so it attracts water, right? That's seen in both of these. Now, which increases the permeability of the hydrophobic region? Basically, do you see a kink in the tail? The kink is due to the presence of the unsaturation, like presence of the carbon-carbon double bond, unsaturation basically. That's due to the carbon-carbon double bond. So in Z, even though it's non-polar, it's a hydrophobic region, it's slightly polar, right? So it increases the permeability slightly. That's why the answer should be Z due to the carbon-carbon double bond. 11D. 12. Which statement of collagen is correct? Tissues containing collagen fibers cannot stretch. Wrong. It does have the ability to stretch. Each polypeptide forms an alpha helix. 3 helices are held together by hydrogen bonds. 3 polypeptide chains form a single collagen fiber. No. 3 polypeptide chains form a collagen molecule. Okay, C is the best answer. 3 helices are held together by hydrogen bonds. Each polypeptide chain forms an alpha helix. That's not right. Okay, that's not right. The structure is helical in general. 3 combine together to form the triple helix, which is held together by hydrogen bonds. That's the best answer. 13. Catechol is a chemical found in a number of fruits. Catechol can be oxidized to quinone by the enzyme catechol oxidase. Catechol oxidase is inhibited by para-hydroxy benzoic acid (PHBA), which structure is similar to catechol. In the presence of PHBA, the mechanism maintained for constant for catechol oxidase increases. So the value of Km increasing, that means that affinity actually decreases. Okay, so PHBA and catechol can both bind to the enzyme, but not at the same time. PHBA is a non-competitive inhibitor of catechol oxidase. It is structurally similar. Okay, so it is supposed to be a competitive inhibitor. Could I clarify? It's supposed to be a competitive inhibitor, so it does not bind to the allosteric side, it actually binds to the active side. So yes, they can bind to the same site, the active side, but not at the same time. 1 is correct. 2 is not correct because it has the same structure, it binds to the same active site. 2 is wrong, and 1 is correct, so it has to be B. PHBA decreases the Vmax. We don't know that. No, it doesn't. Remember, suppose this was the typical graph. So what does a competitive inhibitor do? It actually keeps the Vmax the same. Okay, Km, the mechanism maintain constant increases, but Vmax remains the same for competitive inhibition. But for non-competitive inhibition, what happens is that half Vmax, the Vmax changes. Okay, the Vmax itself changes. It doesn't remain the same anymore. Okay, the Vmax changes, it doesn't remain the same anymore, but Km remains more or less the same. Km remains more or less the same, remains in the same spot. Okay, that's the interesting part. So we don't know about 3. Okay, now 14. Which statements about the fluid mosaic model of a membrane are correct? Channel proteins are fixed in position. Clearly 3 is wrong. They are always fluid. Okay, so 3 is wrong. So we have our answer. It's B. Glycoproteins in the outer layer can move through. The movement of phospholipids is reduced to the fatty acid till they're saturated. True. Less fluid. 15. Diagram 1 and 2 show how the transverse section through a leaf changes when the leaf is moved from solution X to solution Y. Which row describes the water potential of the leaf cells and surrounding solutions in diagram 2 compared to diagram 1? Basically, you need to understand something, guys. What's the logic? Water potential of these cells in diagram 2 compared to water potential of these cells in diagram 1. Okay. Water potential of solution Y compared to water pollution, water potential of solution X. Basically, we need to look at turgidity. The answer should logically be A. I'll tell you why. So, water potential of leaf cells in diagram 2. Okay. These leaf cells have lower water potential compared to the leaf cells in diagram 1. Why is that? Water has basically entered the cell and it has become more turgid. In diagram 2, the leaf is more turgid comparatively. That's why it's firm. Okay, firm in shape and stretched out like that. Also, water potential of solution Y must also be more negative compared to solution X, right? Or else water wouldn't have entered, more water wouldn't have entered into the leaf and it wouldn't have been so firm and rigid and turgid, right? That's the logic. So, both the solution, right, solution Y is more, has a more positive water potential and the leaf cells in diagram 2 has a more positive water potential. Okay, 16. Which features must always be present for water to move between two solutions by osmosis? Okay, we need a partially permeable membrane, that's for sure. We need 3 and we need a water potential gradient. We don't need these. Okay, we don't need a cell surface membrane, we need a partially permeable membrane or selectively permeable. That's the criteria, not a cell surface membrane. Okay, so 3 and 4 only. 16D. 17. When living pancreatic cells were placed in solution of a red stain called neutral red, the cytoplasm became red. The cells were then removed from the solution of neutral red. The red stain in the cytoplasm moved into vesicles, which were exported from the cell, eventually leaving the cell colorless. Which transport mechanisms could explain how the red stain entered and left the cells? Okay, interesting. We use living pancreatic cells. Okay. Now, cytoplasm became red because red moved into the cytoplasm. The cells were then removed from the solution of neutral red. The red stain in the cytoplasm moved into vesicles which were exported from the cells, eventually leaving the cell colorless. Which transport mechanisms could explain how the red stain entered and left the cell? So remember guys, leaving the cell is quite easy because when you move in vesicles out of the cell, that process is called exocytosis. We're going to look for exocytosis. Now, what about coming into the cell? Remember that osmosis only applies to water. Did water move into the cell? No, this was just a dye, so it should be diffusion. Simple diffusion. 17b. 18. DNA molecules in a eukaryotic cell must be packaged compactly because their combined length is much greater than the diameter of the nucleus. Which structures are required in the process of packaging DNA molecules compactly? So remember, packaging DNA, we need histone proteins. DNA is wrapped around histone proteins like this. Okay, those are the histone proteins. They are basic in nature, right? 19. Which statements about mitosis are correct? Mitosis results in genetically identical cells. True. It helps in asexual reproduction. That's true. Mitosis occurs immediately after cytokinesis. No, we have interphase. 3 is wrong. 3 is wrong. Mitosis can be used to repair damaged cells. No, it's used for tissue repair, not cell repair. Could I clarify? So 19 is C. 4 is wrong. A human chromosome is made up of different parts. Some parts of a typical human chromosome are more numerous than others. Which parts are listed in order from least numerous to most numerous in human white blood cells? Okay. So look at this, guys. This is a chromosome. For each chromosome, there are 4 telomeres. Okay, we have 2 chromatids. Okay, and there are plenty of histone proteins, like lots, uncountable. Okay, so, and where's the centromere located at? We have a single centromere at the middle. So 1 centromere, 2 chromatids, a lot of histone proteins. A looks good. B, why is B wrong? Because DNA molecules, we have 1 DNA molecule here, 1 here, 2 in total. We have 4 telomeres, 2 DNA molecules, 4 telomeres, 1 centromere. Wrong. We have multiple histone proteins, 4 telomeres. Wrong. Telomeres 4, centromere 1. Wrong. Get it? 21. A piece of double-stranded DNA containing 12 into 10 to the power 3 nucleotides is transcribed and translated to produce a single polypeptide. What is the maximum number of amino acids in this polypeptide? Okay, guys, triplet codons. Each, like, 3 nucleotides or a triplet codes for 1 amino acid. Okay, so 12 into 10 to the power 3 divided by 3. How much is that? 4 into 10 to the power 3 triplets. Okay, so 4 into 10 power 3 should be the answer. But logically, hear me out. This is actually double-stranded DNA. It's double-stranded DNA. Remember, for DNA transcription, protein synthesis, we only use one strand. Okay, so in total, there are 12 into 10 power 3 nucleotides in 2 strands. So in one strand, there should be 12 into 10 power 3 divided by 2, which is 6 into 10 to the power 3. Now, it's halved. Now, to find out the number of triplets, 6 into 10 power 3 divided by 3 is 2 into 10 power 3 amino acids. This should be the correct answer. 21 should be C. Get it? Now for 22, which statements about complementary base pairing are correct? Cytosine forms 2 hydrogen bonds with guanine. Wrong. It forms 3. 1 is wrong. Our answer is D. So this is how you save time, guys. This is how I used to solve MCQs back in the day. Okay. I actually gave my exam, AS exam, back in 2017 and A2 in 2018. I had country highest in A2 bio. Okay, I got 95 in AS and A2. So you just have to save time. Like while giving the exam, you never know when, like, you'll have a shortage of time for sure. So make sure you just save time. I'm going to show you the others too. Purines and pyrimidines are different sizes. Yes, true. This is double ring. This is single ring. Adenine forms the same number of hydrogen bonds with thymine as it does with GSO. True. 2 hydrogen bonds. The base pairs in a molecule of DNA are of equal length and equal width. That's true. Purines to pyrimidines. That's true. So 2, 3, and 4 are correct. So 22 is D. 23. What are the products when a DNA molecule replicates? This is question number 23. Two molecules of DNA, each made of a sequence of paired bases. Two molecules of DNA, each made of a sequence of paired nucleotides. Two strands of DNA, each made of a sequence of paired bases. Okay, so basically, what are the products when a DNA molecule replicates? The DNA is already double stranded. When it replicates, we get this according to semi-conservative replication. We had 2 strands initially and 1 DNA molecule. Okay, now we have 4 strands and 2 DNA molecules. That's why C and D are wrong. We aren't getting 2 strands, we are getting 4 strands in total. Okay, we're getting 4 strands in total and 2 molecules of DNA. Okay, each made of a sequence of paired nucleotides. B is the best answer. 23B. 24. Which descriptions apply to phloem sieve tube elements and to xylem vessel elements? No cytoplasm. There's no cytoplasm in xylem, but there is cytoplasm in sieve tube. So 1 is not correct. No end walls. No end walls. No nucleus. Okay, both of them, like this one is dead. It doesn't have a nucleus. The sieve tube element does not have a nucleus either. Okay, so this is true for both of them. Now about 2. About 2. Okay, so what's an end wall? End walls basically, xylem does not have end walls, but remember, phloem does have end walls because in between two phloem, do you know, there's this sieve plate. If you view it from above, it looks like this. There are a lot of pores. It's modified into some perforation plates. Basically, there are a lot of holes in it. These are called the sieve tubes, the pores, right? So, xylem vessel does not have a nucleus. Okay, sorry, it does not have an end wall, but phloem does have a modified end wall. So 2 is also wrong. It should be D. 25. Some of the parts of a plant root involved in water transfer are listed below. Which sequence shows part of the pathway through which water can move across a root? Water cannot pass through the Caspian strip, remember that, due to the seaborne deposits. Okay, so what's the pathway actually? first, in the exterior, right? What do we have? What do we have in the exterior? The endodermis is directed in the center, and the epidermis is directed towards the outside, basically. So that's outside, right? Then we have the cortex, and then we have the endodermis over here. So epidermis, cortex, endodermis. So logically, it should be 4 first, the epidermal cells, followed by the cortex cells, followed by the endodermal cells. We cannot pass through the Caspian strip, followed by the xylem vessels. So let's match this. Do we have 4, 2, 3, 5? We don't. And anything with 1 is wrong, basically. Okay, so we have this 2, 3, 5. This matches. A is the correct answer, and this is wrong. It should have been 4, 2, 3, not 4, 3, 2. That's how you answer. 26. What is an example of a mass flow system in a plant? Okay, interesting. The loading of amino acids from the surrounding tissues into no. The movement of sap through the phloem in the stem. This is true because the hydrostatic pressure builds up. Okay, but the first one is an example of secondary active transport or loading basically. Okay, it's not mass flow. And the movement in the root is along the symplastic pathway is not mass flow. Movement of water vapor from the internal spaces is not. But the transpiration stream is an example of mass flow. It's not in the options though. 27. The diameter of a tree trunk usually decreases slightly during the day. Which changes in environmental factors during the day could cause the diameter to decrease even more? So I just did this in my previous variant, October November 2021 variant 1 3, I guess. So where due to the transpiration flow, due to the capillarity action, the diameter of the tree trunk actually decreases. So we want to see which factors increase transpiration. This one increases transpiration, this one does not, this one increases transpiration. So it has to be A logically. 27 is A, okay? Because both of these increase the steepness of the water potential gradient. 28. Which statement about the role of co-transporter proteins in the movement of sucrose is correct? Co-transporter proteins pump hydrogen ions into phloem sieve tubes along with sucrose. Wrong. Wait. Wait. So it's like this. I'm going to tell you the whole process. This is the sieve tube element, and this is our companion cell. This is the companion cell wall. Initially, we pump H plus ions using energy into the cell wall of the companion cell from the companion cell. Then it flows down along with sucrose. The proton that's built up, the proton gradient that's built up, protons flow down the potential gradient through a co-transporter carrying sucrose along with it. Okay, so it's a passive process. That's why this word is wrong. Co-transport proteins pump hydrogen ions into phloem sieve tubes. No. The pump is a wrong word. Basically, co-transporters, help in the movement of protons and sucrose into the companion cell, not the phloem sieve tube. Next, hydrogen ions diffuse through co-transporter proteins. Yes, diffuse is the word. With sucrose into companion cells. Perfect. It's facilitated diffusion, not active transport. B is correct. Okay. This is wrong. Plasmodesmata does not require co-transporters. Passive process. Which row correctly describes the heart during atrial systole? Okay, during atrial systole, blood goes from the atrium to the ventricle. So the walls of the ventricle should logically be relaxed because it's getting blood. The atrioventricular valve should be open. Okay, so 29 is D, and the similar valve should be closed. Easy. During the diastole or the ventricular systole, the similar valve should be open and the atrioventricular valve should be closed. 30. The diagram shows a capillary, some tissue cells, and a lymph vessel. Which row correctly identifies 1, 2, 3, 4? 1. Do you see this by this disc? This is basically your RBC. Okay. So what is 1? From RBC, obviously oxygen should diffuse out, right? Now what is 2? What else goes from the blood plasma into the tissue fluid? Water and small solutes or water and plasma proteins? Think about it. Plasma proteins are actually too big to pass through, so it should be C. Carbon dioxide goes from cells to plasma, okay. And what about 4? It's the flow of tissue fluid into the lymph vessels, right? 31. The table shows the results used to draw an oxygen dissociation curve for adult hemoglobin. What is the percentage increase in volume of oxygen per gram of hemoglobin when the partial pressure increases from 3 to 5? Okay, so it's 0.96 minus 0.58 divided by original 0.58 into 100%, right? So 0.96 minus 0.58 divided by 0.58 into 100%. That's an increase of 65.5% of oxygen per gram of hemoglobin. So 31 should logically be D. Great. 32. The graph shows the oxygen dissociation curve for adult hemoglobin at 2 carbon dioxide concentrations. This is the Bohr shift, it goes to the right, which letters show measurements taken in the capillaries of the lungs and in the capillaries of active muscle. Okay, so basically remember, in active muscle, the percentage saturation of oxygen is low. It's between S and R, okay. But due to the high carbon dioxide concentration in muscles, Bohr shift occurs, and the curve, we need to consider the curve on the right hand side. So active muscle should be R for sure, okay. Now what about the lungs? In the lungs, the partial pressure of oxygen is higher and carbon dioxide is lower, so we should be considering the curve on the left. Bohr shift does not occur. And in the capillaries of the lungs, saturation should be high because we are loading oxygen into hemoglobin, okay. So logically, 32 in 32 in lungs, it should be at P, okay. So it's going to be P and R. That's the logic, okay. So this is in the lungs and this is in active muscle or tissue or anything, okay. So remember this, guys, it's really important. And S and Q are almost impossible instances, okay. 33. Which substance can combine with adult hemoglobin and reduce the oxygen carrying capacity of the blood? Okay, carbon monoxide can do that irreversibly. Carbon monoxide irreversibly binds to hemoglobin. Now, hemoglobin also binds to hydrogen ions to form hemoglobinic acid, causing the Bohr shift, right? And carbon dioxide also binds to form carbamino hemoglobin. Okay, so it's actually 1, 2, and 4. Hydrogen carbonate ions do not combine with hemoglobin, so 3 is excluded. 1, 2, and 4. 33B. Next, 34. This is a tricky question and it's a bit contradictory in a few cases. It varies from MCQ to MCQ. How many times must a molecule of carbon dioxide pass through a cell surface membrane as it diffuses from an air space in an alveolus through a cell in the capillary wall into a RBC? The answer is 5. I'm going to show you. Look at this. Why don't I bring a picture for you guys? Look at this, okay? Wait, how do I do this again? Yeah. Check this. So, where are we located? In the alveolus. We want to go to the RBC. Okay, so this is the alveolus. So let's go. Cell surface membranes. How many do we need to cross? Okay, so I'm going from here into here. Okay, so here we go. Cell membrane 1, cell membrane 2. Okay, so endothelium, the squamous epithelium of alveoli, we cross it 2 times. Okay, then third one, this is the capillary endothelium, one side. Now capillary endothelium the other side, 4. And then into the RBC, this is the 5th cell surface membrane that we are crossing. So it's like this, okay. So for squamous epithelium twice, then the capillary endothelium twice, then the RBC membrane once. Get it? Okay. So there are variations of this question, so remember to do it carefully. 35. Almost at the end. The heart rate of a person at rest was recorded at intervals of 2 15-minute periods. During the first 5 minutes of the second 15-minute period, the person smoked a cigarette. So we don't have questions on cigarettes anymore, okay? So you can skip 35, and 36 can also be skipped because we do not have COPD anymore, okay? Emphysema, then bronchitis, right? So COPD is chronic obstructive pulmonary disorders, pulmonary disease, okay. So we do not have those, bronchitis and emphysema. Measles. We do not have measles, but the basic idea is important. Which factors contribute to outbreaks of measles after natural disasters? So measles does not. You don't need this really. I'm just doing it. Measles is not, transmitted in drinking water or untreated sewage. That would be for cholera. So one is basically wrong. We should go with D. Overcrowded accommodation, true, and lack of effective vaccination, that's true. 38. The two main classes of antibiotics, broad spectrum and narrow spectrum. Broad spectrum antibiotics affect a wide range of bacteria, while narrow spectrum antibiotics affect specific types of bacteria. Which uses of antibiotics may result in antibiotic resistant bacteria? So, a broad spectrum antibiotic given to reduce the effects of the influenza virus. Okay, we should not do that. A broad spectrum antibiotic to treat Mycobacterium tuberculosis in humans, and narrow spectrum antibiotic to treat diarrhea and sickness in cattle. So basically, diarrhea may not be due to it might have a viral cause. Okay, it might not be due to bacteria. Basically, a narrow spectrum antibiotic should only be used when you're confirmed that you know the case. For example, if you know that it's TB, you can use a narrow spectrum antibiotic to counteract that basically. So, right, so a narrow spectrum antibiotic should be used when you're sure about the disease agent, not in something like treating diarrhea and sickness in cattle. That way they can develop resistance. Also, broad spectrum antibiotics in general should be given when you're not sure what the virus is. Like broad spectrum antibiotic may have been right in 3 but not in 1 and 2. We should have used a narrow spectrum antibiotic to kill all the tuberculosis bacterium, right? So yeah, all 3 of these will contribute to antibiotic resistance, because a broad spectrum antibiotic may not kill the tuberculosis bacterium fully. Some of them may survive. Same goes for influenza. Okay, 39. Autoimmune diseases not in the syllabus anymore. 40. Why are spleen cells fused with myeloma cells during monoclonal antibody production? Okay, so basically, what do we do to form the hybridoma cells? We fuse cancer cells and the lymphocytes, basically, right, to get the hybridoma cells, the clones. So why are spleen cells fused with myeloma? So why do we use myeloma, the cancer cells? To give them immortality so that they have uncontrolled mitosis, basically. Okay, so that they can reproduce indefinitely. That is the purpose. This is the cancer cell, basically. Okay, we do that so that they have immortality, because lymphocytes would die in a few days then, right? That was the purpose of doing this ultimately. Do you understand? Okay, guys, that's it. So I'm gonna do the May June series for bio very soon, 2021, within 1 to 2 days, and then I'm hoping to do the specimen papers for biophysics and chem. I'm taking other requests though, but I'm not sure if I can do them within this week. I'll try my best. Okay, so good luck to you guys anyway. So I'm gonna link the playlist for bio paper 1 up here, and I'm gonna link the paper for um, Feb March 2022 paper 2 here, and Feb March 2022 paper 1 2 down here. Okay, and remember to subscribe to the channel. See you for the next one.